what comments can we make about this? What's interesting about this? How might this relate to some of the work we've done in the energy unit and some of the work we're about to do, maybe? Any suggestions from anyone? Apparently this jar was set up in the late 1960s, and it is indeed self-sustaining, self-contained. So one thing, it's been watered once since the 1960s. And there was water in it originally, and it was watered a second time. And therefore, that's an ecosystem, even though a small one, because it's self-contained. Within the jar is everything it needs. So I find that absolutely extraordinary. So let's just have a look. 1960. Put some compost water and plant seeds into a large glass jar and sealed it up. And it's been growing like that ever since, thriving in its own self-contained. The fact it's self-contained says it's an ecosystem. Except for when he opened the bottle in 1972 to water it. What we're moving towards now is this idea that nature recycles. Okay, so think about that. First of all, it is in the light, it's not in the dark, so it gets light, so it makes glucose, grows, everything is completely in balance. But there must also be a recycling. For this to happen, there must be a recycling of nutrients. So some of these leaves will, from time to time, die, and they must be completely recycled, go through the roots and turn back to leaves again. So that's an extraordinary sort of illustration of what nature does that perhaps we don't do enough. We don't recycle our products enough. So where we're going today, sorry, you'll see in a minute, is nitrogen cycles, or nitrogen, I like to call it nitrogen recycles, okay? Or don't call it that though. So as an introduction to nitrogen cycles, I want you to, going to give you 30 seconds or a minute to jot down some biological molecules. Okay, think of some biological molecules that we may have studied in AS or at the start of the year. Okay, so let's have a look, folks. Molecules containing carbon, that's pretty much all of them, folks. We are carbon based. Okay, so good. We said starch, we got that. We said cellulose, which is glucose, you know, both of these. Glycogen, the storage polysaccharide in muscles. We said protein. We said fats, lipids, phospholipids. We said DNA, RNA. We added ATP. ATP wasn't there, that's fine. Okay, so next question, which of these contain, well, there's the answer. Which of these contain nitrogen? Okay, because we're doing the nitrogen cycle, folks. So proteins, DNA, and RNA, pretty major component of life. They all contain nitrogen. We also at some point have to do the phosphorus cycle. So that will be phospholipids there and ATP. You can see phosphates pretty damned important as well. However, so this is where we're moving on to, folks, the nitrogen cycle and the phosphorus cycle. I'm afraid, you know, there's probably quite a lot of listening to do. I do apologize for, but it's the only way now. It's a new topic, if you like, new subtopic. So we have this, I'd like you to think, so nature recycles, and so the nitrogen, you've got the nitrogen cycle, the carbon cycle, the phosphorus cycle. And so what happens when we look at life, when we look at the ecology, we see inorganic molecules, which you can class as abiotic, become biotic. So non-living molecules become living. Okay, and so the non-living molecules are very simple. H2O, O2, CO2, nitrate there, NO2 minus, nitrogen, ammonia, sulfate, phosphate. Those are the bulk of the inorganic molecules. Okay, I'll show you how this breaks down. And producers, plants, turn them into complex molecules such as carbohydrates, proteins, lipids, nucleic acids, DNAs, essentially. So that's far more complex. Okay. Now, by mass, carbon, hydrogen, and oxygen are 99% of the mass. Okay. But the elements, next 0.5% of the mass would be things like nitrogen, sulfur, phosphorus, potassium, calcium, and magnesium. Okay, and then there'll be a load of trace elements that are a very small amount by mass. And we're not going to study them as such, but they are there. Okay, they're not really on the course. You don't need to worry about those too much. So I'm doing another what's the story here. Let's throw this out to you. So this is plant peas. Okay, peas, and how many pods of peas we are getting. And this is the yield. All right. And so the first crop was grown and we added nitrogen, or nitrate to be more accurate. We added nitrate fertilizer, and 0 equals without adding any nitrate. So we added nitrate, we got this. We added nitrate and phosphate and we got that, and we had nitrate, phosphate, potassium, we got that. So could we sort of explain what's happened here? So again, I'll just give you a little minute to think about it. A simple and more detailed explanation. Simple explanation, we've added some fertilizer to these pea plants, okay? Possibly a more detailed and complex explanation. Those of you who I taught chemistry to last year, which is only Samra, I think. She's not here. Samra not here. Samra. I've seen this slide before. So I used it in GCSE Chem when talking about fertilizers. Okay, George, what do you think? What's sorry, carry on.
Because you're adding like molecules that are needed to make other like biological molecules, they can then be used for more growth.
Good. So we've increased growth, and it's sort of cumulative. Could you add to, when we're adding the nitrogen, what sort of molecules are we helping to grow?
Like amino acids, so proteins.
Okay, so phospholipids form the cell membrane. Okay, if you can remember that. And so the cell, if you grow, you need new cells, you need new cell membranes. So that's clear where that is. Potassium, more sodium potassium pumps and stuff like that. So that's one explanation. Okay, nitrogen, amino acids and protein, and DNA for that matter. Phosphorus used to make DNA and RNA, and as said, ATP and phospholipids. Potassium's needed in enzymes in respiration and photosynthesis. Okay, now one surprising thing here though is I just told you that 99% of the plant is carbon, hydrogen, oxygen, and only 0.5% is nitrogen, potassium, phosphorus. But the yield hasn't gone up by 0.5%. So that would suggest, so there's more to it than just, oh, it's been used for growth. Anyone understand where I'm going with that? Because if it was just used for growth, you know, that would be like 99%, and oh, now you can grow now it's 100%, and it will just go up to there, wouldn't it? Okay, we've got more growth. It's actually fairly small amounts of these chemicals relatively that we need. But actually, when we've had these three together, the growth has rocketed. It's absolutely rocketed. So it can't just be the mass incorporating into the amino acids. Can you understand what I'm talking about? So it's not just a pure mass situation. It's also that this is the lack of nitrogen, phosphorus and potassium provides a limit to photosynthesis. And so a lot of this increased mass has occurred because of increased photosynthesis. Can you understand me? It's not because of the phosphorus and potassium itself increases the mass. It's you take the limits. When you add fertilizer to the plants, you're taking the limits off photosynthesis, or one of the limits. You're taking away some of the limiting factors from photosynthesis. You take the limiting factors off photosynthesis. As long as you increase the water, sunlight, growth will rocket. Okay. In general, nitrates and phosphates and potassium are one of the things that can severely limit photosynthesis. All right, and photosynthesis provides biomass through carbohydrate. So that's the other part of this story, is anything that aids with photosynthesis or respiration for that matter is going to be pretty much fundamental to the biomass of the plant. Okay, thanks very much. Now, so let's look at a few things here. So we did mention saprobionts already. We said they were bacteria or fungi, and we said they feed on detritus, which is the remains of dead plants of material and their waste products. Okay. And they perform a sort of recycling function. One of the things we didn't say is, being bacteria and fungus, they do not have a digestive system. So the way they digest is worth mentioning. They would tend to excrete the enzymes into the food, if you like. So the digestion is extracellular, it occurs outside the cell of the fungi or bacteria. And then when those detritus are broken down, then they will absorb it through diffusion, okay, or active transport. So they do not have a digestive system, so they must go around secreting enzymes into the environment, and that is exactly what they do. Okay, and that's saprobiotic nutrition, and they're going to decompose those nitrogenous compounds that we've talked about. Okay, so the nitrogen cycle. In one of nature's great ironies, the majority of the air we breathe is nitrogen, 79%. Most living organisms are unable to use nitrogen gas. Okay, it passes through our lungs, dissolves in our blood without reaction at all, just goes into the lungs, goes straight out of the lungs, even though this nitrogen is a key component of life. We just let it flow through our bodies. Would anyone like to suggest why that happens, or why there's some difficulty, if you like, in what we're going to call fixing the nitrogen? So to get nitrogen from the air into these inorganic compounds, we're going to call that nitrogen fixation. And animals and plants cannot do it. Okay, so despite the abundance of nitrogen, which usually makes for a good chemical reaction when there's an abundance of reactant, we are not able to fix nitrogen. You might have seen in chemistry, both GCSE and A-level, we have this triple bond there, okay? And so the triple bond is considered, despite all those electrons, you thought it might be, I can't remember whether it's susceptible to electrophilic addition or something, but it's considered a very strong bond, okay? And so, well, that's odd, because that's gone not how that slide hasn't gone how I expected. I'll be honest with you, because we can't read it. The enthalpy is 945 kilojoules per mole, so it's very, very strong, as opposed to just a single bond, which is 158. kilojoules per mole. So that's the chemistry reason why we, us humans and all animals cannot fix nitrogen. Okay. So we need to convert it first. Okay. Great. Now, here's a big overview of the nitrogen cycle. Okay, so I'm just going to talk to you about this, and in a minute I'm going to test you on what 1, 2, 3, and 4 are and what those gaps are. Okay. So listen out for that bit. There's a fair amount of talking from me at this point, but I will test you on what we're saying. So this first point we've talked about, we've got nitrogen in the atmosphere, and it's basically inert. It doesn't react with stuff, and I suppose that in itself is useful. I'll just mute most of you in a minute, I think, because it's quite noisy in my ears. Yeah, great. So got the nitrogen fixing bacteria here. There's two types there. You're going to get what's called free living, sounds rather exotic. There's free living bacteria, free living nitrogen fixing bacteria. So what do we mean by free living? What we actually mean there, they're not attached to a plant, because although I said plants cannot fix nitrogen, actually there's a group of plants, and one example of which is the legumes, the leguminous plants, which are your pea plants, which can fix nitrogen only because they have these bacteria in something called root nodules. Okay. And so I'll show you a little bit more about that in a second, but there's a distinction between free living bacteria, which is just bacteria in the soil not connected to a plant that fix nitrogen, and bacteria that attach to pea plants and other nitrogen fixing plants in what's called a symbiosis, a symbiotic relationship. Now, I don't know if you've come across symbiotic relationships before. So a symbiosis is a mutual relationship where both parties benefit. So you are actually already in symbiosis with some of the bacteria in your small intestine, because some of the bacteria in your small intestine are good, actually. In fact, we hope as you're healthy, they all are at the moment, but they're there in your small intestine, and they're getting, some glucose and stuff from your food, but they're also help making vitamins in your food. So they're necessary. They need to be in your small intestine. So they're an example of a symbiosis. Another good one is lichen. Lichen is a symbiosis between algae and fungi. The algae photosynthesize, the fungi protect and provide nutrients. So there's a few examples of cross species actually almost working as one species, okay, together, and that's a symbiotic relationship. Both parties benefit from the alliance, if you like, and so that's nitrogen fixing. I think I do have a couple of more slides on that. Yeah, let's have a look. So the bacteria, the name of the bacteria that do nitrogen fixation are rhizobium. Okay, as we said, they transfer nitrogen to ammonia in the first place, not nitrate. It's worth pointing out here that if you look at this whole cycle, nitrogen fixing bacteria are going to make ammonia. So, by the way, is saprobiotes that we've talked about. Plant root cells only take up nitrate, NO3 minus. Okay, so the ultimate goal of the nitrogen cycle really is to form nitrate NO3 minus, because NO3 minus is absorbed freely by all plants through their roots by active transport. Okay, so this is just a first step. It leads to ammonia, and you'll see the reasons. The reasons are all to do with energy as to why it needs a number of steps. Okay, we cannot take nitrogen directly make nitrate. Well, we can through lightning, but that's a very high energy random process. Okay, and so it's a mutualistic relationship, symbiosis, so mutual meaning they're mutually beneficial. Okay, so this is a photo of the rhizobium, okay, on the root nodules of pea plants. So these were bacteria that fixed nitrogen. And in the old days, before there was enough fertilizer. So in the old days for fertilizer, they used cow manure, etc. for fertilizer. And then another thing they did was call what they had, they left what was called a fallow field. Okay. Now, if you ever rock up at Manchester University to study, as I did many years ago, you will know there's a whole student area called fallow field. Okay. What it means is that area must have been previously the fallow field. And in the fallow field, you grew only peas and beans, nothing else. And what that was doing was in olden days farming, you'd have 3 large fields. You grow your wheat in 2 of them maybe, and the third one would always be peas and beans, because the peas and beans fix a large amount of nitrogen. They put fertility back in the soil. And then after a year of leaving the field fallow, by growing just peas and beans, you could grow wheat on them again. But if you grew wheat every single year in a field, and then you remove the wheat, you're removing the nitrogen, eventually the wheat crop would collapse. So they had to have a fallow field system. Obviously nowadays we use inorganic fertilizers, and some would argue fertilizers have saved millions and millions of people from starvation, more than anything else. But if you remove nitrogen from the soil, the point is it has to be put back. And so leguminous plants play a role in that. Okay, so where were we on this? So that was nitrogen fixation by a free living bacteria and rhizobium bacteria, sorry, they're called, and by nitrogen fixing plants, which is always a mutual symbiosis between those. Okay, so that's step 1. Now, feeding into that ammonia in the soil is, as I say, the saprobionts. They break down animal protein into ammonia. So let's just have a look if we've got a bit more information about that. No, we've gone the wrong way. Here it is. Yeah. Okay. So proteins, DNA, RNA from all the dead organisms are turned into ammonia by saprobionts, okay, which goes on to form ammonium ions. So ammonia is NH3, ammonium NH4+. Some of you will know that from your chemistry, okay. So all of the destination of all the waste, urine and feces, is to form ammonium first. It's just easier to do. And then, apologies, non-chemists. So those of you that are chemists, you can make the link here. This is an amino acid, obviously. We liberate ammonia there. And what these saprobionts are doing is they're using this 2-carbon molecule there, which looks a lot like vinegar, acetic acid. Okay, so that acetyl, remember that, folks, the acetyl there can go into respiration. They can respire using acetyl. So they are getting, the saprobionts are getting ATP out of amino acids, and they're forming a byproduct, which is NH3, which is allowing nitrate ultimately to be recycled, folks. And that's called ammonification. Ammonification is when you remove ammonia from proteins by decomposers, convert it to ammonium ion. Obviously that's ammonia, that's not an ammonium ion, but it's a pretty small step from ammonia to ammonium ion, to be honest. and they do get something out of it. They get a respiratory substrate. Okay, I don't need to talk about that yet. And then lastly, we have nitrification. Okay, so all that ammonia in the soil will be changed into nitrogen compounds. And you've got two names here, nitrosomonas. They change ammonia into nitrite. Nitrite is going to be NO2 minus, okay? And then nitrobacter are going to turn nitrite into nitrate, NO3 minus. Okay, so the situation there then is even nitrite, NO2 minus, is not uptaken by plant roots. There's only one thing uptaken by plant roots, as far as I know, anywhere, and that's NO3 minus. So we need to convert all of that dead stuff, all of that nitrogen from the air into NO3 minus. Okay, and obviously, if you are doing chemistry, you'll notice we've got oxidation. We've got oxidation going along there. Okay, from NH3 to NO2 to NO3, a whole load of oxidation. We don't need to worry about that in biology anymore. Okay, so why is it done this way? Producing ammonia takes 25 moles of ATP to produce 1 mole of nitrogen. So it's energy intensive to produce ammonia, even for bacteria. And as I say, plants and animals generally cannot do this at all. Fixing 1 gram of nitrogen from the atmosphere costs 10 grams of glucose under favourable conditions. Okay, so it's energetically expensive. Why is it energetically expensive? As Oscar mentioned previously, that nitrogen triple bond, it takes some breaking down, folks, and so there's a lot of energy in that nitrogen bond, and so you can't get something for nothing energy wise. Okay, you're going to have to expend a lot of glucose to get that. Okay. Denitrification is something you don't want. Okay, so that's when you have nitrate in the soil and you have something called denitrifying bacteria this time, and they take nitrates in the soil, carry out respiration and produce nitrogen gas. And this happens when your soil is waterlogged and anaerobic, and you don't want that in general because nitrate is a limiting factor on photosynthesis. So if you've removed nitrate from the soil, then the soil is less fertile, but it can happen. So bear with me, folks. Let me talk through summary of that, and then we'll close. So this is nitrogen fixing bacteria, both free living and attached to nitrogen fixing plants, always to form ammonia. At the same time, saprobionts also produce ammonia from consuming dead plant protein, dead animal protein. Okay. And then, of course, urine, by the way, has NH3 in it already. Okay. So that's the first step, is making ammonia. Then you'll get one type of nitrifying bacteria that will produce nitrites, and the second species of nitrifying bacteria will produce nitrate. Nitrate will be uptaken by the roots. Only one problem: if the soil is waterlogged, there's a different group of bacteria, denitrifying bacteria, which will turn the nitrate into nitrogen gas. Okay, there. So the 5 processes we've looked at are nitrogen fixation from the air, nitrification, which is turning ammonia into nitrate. Assimilation, is the word. Assimilation is when that simple, is what we talked about at the start, simple inorganic molecules are turned into complex organic molecules by a plant. Okay, that's called assimilation. They're taken up. There's a second part to assimilation, is They are assimilated as part of a complex, simple inorganic molecule, complex organic molecule. Okay, that's what we mean by assimilation. Then we've got something called ammonification. That is when the detritus protein is turned from an amino acid into ammonia. The saprobionts tend to use the carbon species there to make ATP, to carry out respiration. Okay, and then last process, what you don't want is you don't want a lot of denitrifying bacteria, because what they do is convert nitrate back into atmospheric nitrogen. Okay, so that's it. We are finished.